distracting to you at all?
Oh, I'm fine.
Okay, cool.
You guys are all here really early. You must not have like any classes right before this, or just have enough time. You can, for now, until you figure it out, you can get like a 2-week free trial, I think, if you sign in through the link on campus. At least do that in the meantime. Okay, so we left off talking about genetics in the news, and I just wanted to give the opportunity if anyone was like, I really wanted to share mine, but we ran out of time. Now's your chance. But zero obligation. I just wanted to open up in case anyone felt like they wanted to. So the next sort of thing I wanted to share is, this is not an endorsement for Netflix technically, but it's just a show. It's like a documentary that is on Netflix. It's like a full mini series, I guess you could call it, that I watched last semester on my own time, and it was really eye-opening for a lot of like current topics in genetics. So I wanted to at least show the trailer just to highlight what sort of hot topics there are, and if you feel drawn to seek out this on Netflix on your own time, that's cool too. So I've never used the sound system in here, so we'll see if I can get the sound to work. Let's see. That's about. Okay.
It's not sustainable.
That's better.
We do not advise that anyone watching this video do what is about to be done here. These compounds are not for human consumption. They are for research purposes only. Our civilization is vulnerable. It's not sustainable. We have no choice but to continue exploring the tree of knowledge. But we always run the risk of discovering something that we cannot handle.
We can take a gene from one organism, put it in another, and make a medication that can save a bunch of people's lives.
It assumed that getting DNA and making changes and stuff cost millions of dollars, and that you needed a huge lab and a research team. You'd be surprised what there is on YouTube.
CRISPR was the starting gun. Lets you edit just about any gene.
Alzheimer's, cystic fibrosis could be diseases of the past.
He's missing both copies of the RPE65 gene.
you will eventually go completely blind.
Then what kind of world can we build?
The risks of humans undermining their own existence are very real.
We are developing gene therapy technologies to help people take the health of their lives into their own hands.
There needs to be rules. Don't put gene drives into the wild.
Gene drives could cause irreversible ecological damage.
It's amazing the things that we could do with this. The question is, should we? What happens if diseases were just eradicated?
Technological breakthrough that could change the future of mankind.
This is truly experimentation on human beings.
These tests can regrow your kidney. Biohackers actively working on it themselves.
How many injections have you given yourself?
We are in control of our destinies now.
If you're really playing God, you don't care what other people think.
So if you didn't catch that, the title was Unnatural Selection. And when I first saw the trailer, I was like, this has to be some like fictional, like futuristic thing. But no, it's an actual documentary that I forget when they started filming it, but it's within the past few years, the world of what genetics is capable of, basically. And they do a good job of explaining the science behind it, but also like the future risks involved in what we're doing or have the potential to do. And I just wanted to sort of throw that out there at the beginning of the course to highlight that we will be talking about some things such as what you saw in that video that are still very controversial, and not everyone in the room will have the same opinion. I'm happy to share my opinion with the caveat that you do your own research and your own opinion forming based on what we learn in class and what you learn outside of class. So a lot of these things that we'll talk about when we get to the more current relevant information as well as future of genetics can be a little bit muddled as far as what's ethically right and wrong. But to get to that current hot topics and futuristic genetics, we really have to start at the basics and learn the history of genetics and the fundamental concepts of genetics before we can really dive in and understand and talk about those controversial issues. So Unit 1, this is all of the chapters that will be on your first examination, and we'll talk about the introduction of genetics today, and then we'll dive into Chapter 2 for the remainder of this week and talk about chromosomes and cellular reproduction and how mitosis and meiosis is involved in passing genetic information on to new cells. And then we'll dive into Mendel and his understanding of heredity and using pea plants as a test to understand how traits are passed from parent to offspring. And then we'll get into, that's more like the basics of heredity, and then we'll talk about how sex is a part of that, and look at characteristics that are linked to sex chromosomes specifically. And then finally using pedigrees as a way to also help predict heredity as well. And part of that is also genetic testing. What capabilities do we have currently for genetic testing of babies or of adults, and anywhere in between. So Chapter 1 is just our really basic foundation intro to genetics, and I'm going to try to show what the learning objectives are for each chapter when we start a new chapter. And basically what these learning objectives are what I hope that you are able to demonstrate to me by the end of this chapter, or by the end of this unit, really. Each of them are based off of one chapter, but really you'll be tested on this at the examination, as well as with your homeworks and the learning curves and things like that. So think of this as a study guide of what to really focus your attention on, because your textbook is really comprehensive. There's a lot in it that we're not going to dive super deep into, so this will really help gear your studying in the right direction. So I'm not going to talk through this. This is just your resource, basically. So for the first part of this chapter, we're basically talking about why is genetics important to this class, to society, to your major, things like that. So a lot of you might be in this class because you're pre-med or pre-health of some sort, and so this aspect of diseases and disorders is going to be directly relevant to that career path that you've chosen. So understanding not every disease is genetically related, but a lot of them have been shown to be influenced either 100% or somewhat by genetics. So understanding the heredity of those genetics is going to be helpful. Agriculture, if you are in a major that deals with agriculture and farming, there's a lot of genetics involved in selecting plants naturally to breed a positive trait that you like, as well as genetically modifying organisms to be bigger. For example, this organism here shown was bred to be bigger to then produce more food for society, for example. Other examples of a modification that they might want to make is to be resistant to herbicides so that they can spray weed killer, kill all the weeds, but the plant remains. Just one other example that it's been used for in agriculture. Biotechnology is going to be things like developing research techniques, and medicine sort of goes along with that. Developing certain drugs that will help, that sort of relates back into our diseases and disorders. But also within medicine, we can use genetic systems from other organisms, such as bacteria, to sort of train the bacteria to make and produce the medicine for us. That's how antibiotics were originally discovered. They had a fungus that actually started producing penicillin naturally. It's a compound that the organism uses to defend against other bacteria that are trying to take up its space. So we can sort of recruit bacteria or fungi to produce medicine for us that'll be beneficial. So understanding the genetic systems of bacteria will help our society as a whole, not just cool to understand bacteria. And then finally, understanding how organisms develop. So we will have a small unit on how genetics is used in the early stages of life development, and how it tells your cells what they need to become and what they need to produce and what proteins need to be turned on, things like that. So hopefully one of those categories interests you to some degree, and this is why you're taking the course. But we will try to cover all of these topics, not just focusing in on human genetics. So a couple points of terminology to get us started. You've probably heard this word genome in other courses potentially. In this course, we will refer to it as a complete set of genetic instructions for any organism. And so for this image here, we have a cell that's zoomed in on maybe a skin cell or something. And within that cell, we have, since it's eukaryotic, we have a nucleus. And within that nucleus are all of the chromosomes that contain the DNA for that organism. So all of the DNA as a whole is considered the genome for this particular organism in question. Not every organism uses DNA. Some bacteria and some viruses use RNA. So we would refer to that RNA genome still if we're referring to the total genetic material for that particular organism. And then the key for heredity is in the fact that this DNA or RNA genome is copied during the process of replication, so that when the cell divides, both cells get a copy of that identical genome and can be passed to the offspring if the cell is being used for that purpose. So for genetics, which is highly related to the study of evolution, we'll touch on evolution towards the end of the semester, but you can sort of be thinking about how genetics relates to evolution all throughout this entire semester, really. But basically, the genetics of using this system of DNA with A's, G's, C's, and T's, that's going to be our coding system that is uniform for all life forms. So even if some life forms are using RNA, the RNA is still the same as the RNA system in your body. This coding system is universal, essentially. So the idea or the theory is that all organisms that are currently on the planet today, since we have a uniform system, it's most likely that we all evolved from a common ancestor that had that genetic system, and we've diverged as DNA has mutated slowly over time. And finally, we wouldn't have evolutionary change if it wasn't for mutations to the DNA causing a variety of genetic variation. So everyone in this room looks different. We have really, really similar DNA, but we have slight mutations between us that code for different proteins, and the outcome of that protein produces a different phenotype. That's why we all look different. But we all share that same genetic system. Okay, so our next sort of terminology key here is the central dogma. Again, might be a familiar term from biology, but does anyone want to shout out the 3 sort of terms that come up with this central dogma?
DNA, RNA, protein.
DNA, RNA, protein. So, and that's in the right order even. So we start with DNA if it's an organism that has a DNA genome. And this is double-stranded, right? So think of it as 2 strands connected, and it's complementary. And then, what was the next one?
RNA.
RNA. How is that different from DNA?
T's are used, and it's going to be complementary to the DNA.
Okay. And is it double-stranded or single-stranded typically?
Single-stranded.
Typically single-stranded. So in this case, we have A's, G's, C's, and T's. In the case of RNA, T's are now U's, and we have a single-stranded nucleic acid that was complementary to this original DNA. So it's sort of a message in order to make what's our final goal of the central dogma. What was the last one? Protein. So RNA is used to make protein, which is made up of a string of amino acids. And the code for what order of amino acids is going to come from the sequence of the RNA. And that'll tell the ribosome what protein to make. So we've got our DNA here with A's, T's, G's, and C's. The RNA is the same as the DNA, except the T's are now used, and it's single-stranded. And these, in triplets, called codons, will be used to translate the RNA into protein. So this is methionine. We'll get into the really specific details of this later on in Unit 2. But this is the central dogma of transcription. Oop, went the wrong way. Transcription being used to make RNA from DNA, and translation being used to make a protein polypeptide from mRNA. So we've got a little definition down here that this is the central dogma of molecular biology is describing that 2-step process. The first step being transcription, the second step being translation. And the flow of information goes from DNA, RNA to protein. Peptide is just another word for protein. Okay, so within our subject of genetics, there's 3 different types of categories that genetics information might fall into. So the first being transmission genetics. That's going to be sort of what we dive into first because it was historically the first one sort of looked into. So we also refer to this as the more classical genetics, where we're just looking at the physical traits that we can see, either fur color, or when we talk about Mendel, we'll talk about pea plants and the different phenotypes like round seeds versus wrinkled seeds. And looking at the transmission of those traits, we can start to piece together some theories of how genetic inheritance patterns work. So we'll talk about the history of that and the facts that we know today. And then the other sort of realm of genetics is molecular genetics. Clearly, it took a little bit longer to understand how molecular genetics works because we didn't have microscopes early on, and we didn't have ways to sequence DNA. We didn't know what a protein was and how it was made from DNA as the information flowed. So as historically we understood these things more and more, we started to build this molecular genetics, and we can start to understand, okay, we understand how things are inherited, but why does black fur look different than white fur? What genetically is different? And that's where molecular genetics comes in, to sort of answer the why rather than the what. And then finally, this third realm is population genetics. And that's more looking at groups of individuals that share a pool of similar traits and where they're moving to and how they're interacting with each other. So that falls under the category of population genetics. Okay, so let's check in, see where we're at, make sure we're all staying awake. Think to yourself, if you were a scientist studying a newly found gene, you're working to discover the structure of the gene. So we haven't really talked about genes yet, but a gene is a piece of DNA that codes for a certain trait. So if you're looking at that gene and you're looking at the product that it expresses, which is our protein, this would be our product expressed from the gene. You want to also know the function of that product. What is that protein doing in the body? What subdiscipline of genetics would this fall into? Molecular? Why do you say that?
Because it's more or less asking the why, so it's asking why it is being expressed because of the way the protein is formed.
Right. And so understanding genes and the product and what the product does, that's going to require an understanding of the molecular nature of genetics, rather than just looking at the transmission of that gene. Yeah. Okay. How about this one? Which of the following subdisciplines of genetics focuses on how an individual organism inherits and passes on its genes to the next generation?
Transmission genetics.
And why do you say that?
Because that's the definition of it.
Okay. Why not population genetics?
Because it's not necessarily talking about big groups, it's talking about individuals.
Right, exactly. So if it were talking about the inheritance patterns of large groups, you might think population genetics. But in this case, we're talking about an individual organism and how it's transmitting from one generation to the next. And the key there is looking at a parent generation and an offspring, and then possibly further offspring. Whereas population genetics may or may not look at different generations. Okay, so our next topic to discuss is model genetic organisms. So not everything can be studied in humans because humans are a difficult research subject to get approval for, right? There's a lot more hoops and red tape to get through. So a lot of times, since we have this universal genetic system that works the same, we have this DNA to RNA to protein in every organism on this planet, including plants and bacteria. So we can use model organisms to study a protein or a gene and its outcome by investigating it in an organism that's a lot easier to work with in the laboratory setting. So these model genetic organisms have some sort of characteristic that makes them useful to study genetics. And there's no perfect model organism. It just depends on what your research question is. But some key features that make a model organism more attractive to use is a short generation time. So you're going to have parent to offspring to second generation to third generation much more rapidly than, say, horses if you're using fruit flies. I don't know the exact replication generation time, but I think it's less than a week for them to have offspring. So that's going to be a lot easier to look at transmission genetics if you're studying that versus a horse that has a much longer gestation period. A product of numerous progeny, so that indicates that they are having a large number of offspring. So how many offspring can a horse have at once? Anyone know? One. Has there ever been an example where there's like 5? Probably not. And if it is, it's super rare. Whereas fruit flies and all these other model organisms listed here have a high number of offspring. And so the higher your number, the more statistically significant you can draw for your conclusions, right? So you can get much higher amounts of data if you use one of these model organisms. The ability to carry out controlled genetic crosses, basically saying you control who the parents are and who they're mating with, so you know exactly, okay, if this trait is being mated with this trait and this is our outcome, it was a very controlled experiment. They weren't just randomly mating, and you can then track certain traits in the lab. The ability to be reared in a laboratory. So all of these examples are things that you could contain within a lab and control. That way it's under a controlled environment. Climate is controlled. All of the resources that it's available to is controlled, things like that. Yeah.
Is like marine biology tougher because some animals can't like live in a lab? Like
Right.
I've heard like great white sharks, like they can't stand in a tank.
Yeah.
Are there some like realms of like like marine biology that are tougher because of genetics that would be like?
Right. And that goes into, you choose your model organism based on your research question. So if they were studying a behavior in that particular organism, you can't study that in any other model organism. It would have to be studied in the wild or whatever. If it was a protein that's involved in the metabolism of something in that organism, that might be able to be studied in another organism that also uses a similar protein. So it just depends on what the question is. But yeah, if your research question is complicated in the fact that you have to use that organism, it's going to cause some challenges that you have to overcome. Yeah. And then, of course, the last one, you have to have a knowledge of their genetic system. So you can't study the genetics of E. coli, a bacteria, if you don't know anything about how their genetic system works. So most, if not all, genetic model organisms have their entire genome already sequenced. So we can just pull it up on a computer and say, I want to look at this gene, I want to mutate this gene to this and see what happens and see how that affects glucose metabolism, for example. You can't do that unless you have a knowledge of the entire genome. It makes it a lot more complicated and difficult if you don't already have that information. So that's what makes a good model organism. Okay, so let's check in. Which of the following are characteristics of a model genetic organism based on what we just talked about? Would a slow generation time be useful? No. We would prefer it to be a faster turnover. What about easily defined characteristics? Yeah, that would be really helpful, right? What about a small number of progeny? No. The higher number, the more data points you have, so that's a benefit. Requiring highly technical equipment to be studied, something you want to avoid, but some research questions would require that, so it just depends. But you want to try to avoid that if possible. And then easily studied outdoors. It would be much more useful if it was able to be studied in the lab environment. But of course, it all depends on what the realm of possibilities is. So if ever possible, you want to study it in the controlled lab environment. So the key here is having easily defined characteristics, especially if you're looking at the transmission of those characteristics. Okay, which of the following would not be a model genetic organism? Budding yeast, do you think that, which is a small fungus, do you think that would be something that you could work with in the lab and quick generation time? Fruit fly, we already talked about that being a good model organism. Human beings, not ideal. Of course, some questions have to be answered with humans, but ideally, if we could take the genetic question that we're trying to answer with humans and put it into fruit flies or E. coli or worms, that would be ideal. Worms and any sort of plant is typically going to be a good model organism as well. So humans, not ideal. Okay, so the second section of this chapter is basically a brief history of genetics, and I've boiled it down to what was proposed as a theory and whether or not it was supported by genetic experiments or not. Because I don't want you to get bogged down with names and dates and what the experiment was. As we go into future chapters, I will draw on specific experiments and specific people, but for this early foundation, I just want you to sort of be familiar with what sort of concepts were wrestled with and what do we know to be true today. So the first one was that they thought, when they were trying to figure out how is our relatedness sort of passed on from one generation to the next, we're noticing that our offspring looks a lot like our parents, but we don't know why or how. So one of the theories was that genetic information from all parts of your body will travel to the reproductive organs to be passed on to the offspring. So all the information for your eye color and your hair color and your skin type is going to be passed. All of that information travels to the cell that's going to be used for reproduction. We know that is not true today, and that instead this germplasm theory is what we support today, in that all cells in your body contain that complete genome. All of the genetic information will be stored within that cell, but not every cell expresses every single gene. That's why your eye cells behave different from your skin cells. But all of your cells, including your reproductive cells, the gametes, those also contain all of the DNA. It's not collected from all different sections of the body. This other theory that early geneticists, before it was even called genetics, had was that acquired traits would be incorporated into hereditary information. And that was pretty easy to disprove. You've probably heard, what did I press? I don't know what I pressed. I don't think this controls that at all. What'd you say?
So it looks like class is over.
Yeah. I don't know. The computer itself turned off. Oh, it's back on. I don't know what that was. Sorry. What was I saying? Oh yeah, acquired traits being inherited was pretty easily disproved. If someone lost their arm, it doesn't mean that their offspring is also not going to have an arm, things like that. So that was easily disproved. And then the next sort of theory that someone had was that these teeny tiny miniature organisms reside within the gamete sex cells, and that all traits are inherited from one parent. So they thought within that teeny tiny sperm cell there's a miniature organism that has everything that it needs, and it's just waiting to have the nutrients from the mother's egg, and that then it will grow. But we disproved that after some time, and we know that now half of your genetic information comes from the father and half comes from the mother after the two cells merge. Another theory that was disproved was that genes blend and mix. So the idea was that if you have a black fur cat and an orange fur cat, they merge together and would have a brown fur cat, something like that. But now we know that genes aren't that simple. Each individual gene ultimately is coding for a protein. And if that protein is on, one thing will happen. If the protein is off, a different thing will happen. Or if the gene is mutated slightly differently, you're going to get a slightly different protein. So it's not the fact that your dad's gene mixes halfway with your mom's gene. It's all or nothing. You're either going to express the one from your dad or the one from your mom. But you're not going to express both at the same time. Let's see here. All life is composed of cells, and cells only arise from other cells. So that is our cell theory, the idea that you can't get something from nothing. All of your, when life is first formed, it's coming from other cells to produce those cells in the first place. So we'll talk about mitosis, in which cell division goes from one cell and that splits into two daughter cells. And then when we talk about fertilization of the sperm and the egg, that's going to be another sort of type of making a new cell. And then finally, Mendelian inheritance is going to be what we talk about, chapter 3, I believe, is all about Mendel and his experiments with the pea plants and all of these defined principles that he gathered by looking at what traits showed up in what generation, at what ratio, and things like that. So that's sort of a brief overview of this long history of genetics. So I showed you this on the first day of class, and just again to reiterate, there's a lot of people that have been involved in our current understanding of genetics today, and I don't want you to memorize all the names and dates and experiments, but I do want you to have sort of like this broad understanding of what was proposed, what do we know is true today based on experiments that have been performed. Okay, so the last section is basically a brief overview of all the fundamental concepts that are going to be really important to have a strong grasp on before we get into all the future chapters. So I'll sort of highlight them here, but I really encourage you to read that section in your book. It's not very long, and it just sort of elaborates on what I have up here. So first, you need to either remind yourself or relearn that eukaryotic cells are unique from prokaryotic cells. Even though they share a similar genetic system, they still have DNA to RNA to protein. They have very unique characteristics. Eukaryotes are what? Are we eukaryotes or prokaryotes? We're eukaryotes. We have nucleus, we have organelles, things like that. So therefore, they're going to behave slightly different with how their genetic information is processed and packaged and transferred to new cells and things like that. Whereas prokaryotes are things like bacteria and archaea, those more single-celled organisms, and they're going to have sort of a unique system of how their genetic information is maintained. So we have a whole chapter on prokaryotic genetics in the second unit that we'll get into. Otherwise, most of our semester will be focused on eukaryotic genetics. Genes, we talked a little bit about how a gene is that section of DNA that ultimately is going to be expressed into a protein. So these genes are the fundamental unit of heredity. These are the key pieces of DNA that are passed from one generation to the next to pass on the capability to make that protein, which gives the organism certain functions. But these genes are going to be what we can track from one organism to the next. Genes come in multiple forms, which we call alleles. So I mentioned the cat black fur versus the cat orange fur. Those would be the two alleles of the same gene, but they're two different genes. They code for different proteins. So the black fur gene would be one allele, and the alternative form of that, the alternative allele, would be the orange fur. So they're both sort of talking about the same information, but with a different outcome. So we package them and call them alleles. Genes are what confer our phenotypes. So remind yourself of the difference between a genotype and a phenotype. Genotype being the actual DNA sequence of what you have in your cells in the genome, and phenotype being the outward expression of that gene or of that DNA sequence. So ultimately, we have this protein that's made from the DNA, but what does that protein do? Does that protein give you brown hair? Does that protein give you the ability to metabolize lactose? Those are going to be things that are your phenotype, the outcome of that gene. Genetic information is carried in DNA or RNA. Most organisms carry their genome on DNA, although there are a couple examples of organisms that use RNA. Genes are located for eukaryotes and prokaryotes. They both contain chromosomes. They look very different. Eukaryotic chromosomes are the more traditional. You sort of see them packaged into an X shape. A lot of times you'll see a chromosome drawn like that. Whereas prokaryotes, they also have a chromosome, but it's really just a circular DNA. So this is our double-stranded DNA. It's just one big circle. Whereas this is just one chromosome of, how many do we have? 46? 46 chromosomes in eukaryote and for humans, whereas a bacteria just has one single circular chromosome. Either way, the genes, if this is the gene that makes that protein, maybe it's located right here, or on the bacteria, maybe it's this little section here. That's going to be the location of the gene on the chromosome. Chromosomes separate through mitosis and meiosis. Review the general concepts of mitosis, which is cell division from one cell, duplicating the DNA, and separating into two cells. Meiosis is the same general concept, except we go through that process twice to make gamete cells that have half the number of DNA, so that they can go on to be fused with the sperm or the egg to make a final fully formed genome. But ultimately, these chromosomes, if we're talking about human chromosomes, this has to be duplicated. So we make a second copy of it, and then they have to separate. One will go into this cell, and one will go into this cell, in order for both daughter cells to have the same original genome or chromosome. And then we already talked about this a lot. DNA to RNA to protein is that central dogma of information flow. Mutations can cause permanent changes. So if we had an A here, A binds to T. If something came along and maybe UV light mutated that A to a G, now we have a GC, that mutation, if it occurs in a sex cell, that's going to be passed on to the offspring. So that, in a way, is a permanent change that can be inherited, right? Or if this is just in your skin cell, your skin cells are also multiplying and dividing within you, right? So if that mutation happened and caused skin cancer, that is also a permanent change that would have to be, you would want to physically remove those cells that had that genetic change. And then some traits are affected by multiple factors. So when we talk about certain traits and diseases, some diseases are one gene, one protein, the mutated protein causes the disease. Sometimes it's that simple. Other times there's lots of different genes involved, and it's not as clear cut as one gene, one function. So just keep in mind that we will normally focus on one gene, one function, and the outcome, but in a lot of cases, there's multiple factors involved. Not only genes, but also the environment that you're in can influence whether or not a gene ends up being turned on or off, things like that. And finally, we touched on this a little bit, but evolution is genetic change. So if this mutation that we just induced here on the DNA, if that did happen in the sex cell and got passed on to the offspring, if that was a beneficial mutation and it caused a new protein to be made to metabolize a new compound, for example, then that organism would have a benefit over its neighboring organisms and could potentially lead to evolution of that organism in a positive way. And the idea is that over time, little by little, random mutations accumulated in order to diversify our species today. Okay, let's do one last wrap up here. What are some of the implications of all organisms having similar genetic systems? So we mentioned both eukaryotes and prokaryotes share a similar genetic system. So what's an implication of this? Go ahead and turn to your neighbor, see if you guys agree. I hear lots of all of the above. Does anyone have an idea that other than all of the above? Okay, yeah, so all of these are implications of having a uniform system. We talked about the fact that every life form is genetically related. We all, the theory is that we all stem from one common ancestor at one point in time. Research findings of one organism's gene function can be applied to other organisms. So if we study a gene for lactose metabolism in mice, if that protein from that gene is the same as the one in humans, we can relate that study, that research to humans, even though it's not direct. And then genes from one organism can often exist and thrive in another organism, thanks to the fact that this code, this 4-letter code, is the same in every organism. If I took a gene from a mouse and put it into the genome of a rat, the rat would start making whatever protein that gene codes for because it's the same letters. It's not like, oh my gosh, this is a mouse genome. I can't do this. They just see letters and they make RNA from those letters, and that RNA gets coded into protein. It's a really dumb system in a way. It doesn't care what it is or where it's from. It sees the letters, it makes the protein. So that one's all of the above. And then just some announcements of what's coming up. The first 2 learning curves are not graded because I know this is drop add period. There might be new people coming in. You might have trouble accessing Sapling. All those kinks need to be worked out. So feel free to practice chapters 1 and 2.